just saying, hopefully these lights are just above nap level, but having talked for a little bit with Dr. Carroll today, I don't think that's going to be a problem. I could give, the more I learn about Dr. Carroll, the more I realize I could give a whole seminar on him. So I'll keep the introduction kind of brief and quote some of what's just on his website. He's an award-winning scientist. Of course, that's why all of us are here to listen to him. But he's also an award-winning author, educator, and film producer. We had a fun time at lunch hearing a few snippets about some of his upcoming works on PBS, The Gene Doctors. And of course, if you've seen The Inner Fish, you've seen his work. If you haven't seen The Inner Fish, what's the problem? Go see it and watch it over Thanksgiving with your friends and family also. Dr. Carroll leads the Department of Science Education for the Howard Hughes Medical Institute. Importantly, this is the largest private supporter of science education activities in the United States. He's also the Alan Wilson Professor of Molecular Biology and Genetics at the University of Wisconsin. And so then at some point I kind of think he doesn't have time for anything else, but clearly he does, because he's made time for us. And his lab work focuses on the genes that control animal body patterns and that influence the evolution of diversity. He's received numerous awards, the Benjamin Franklin Medal in Life Sciences. He's been elected to the National Academy of Sciences, the American Philosophical Society. He's a fellow of the American Academy of Arts and Sciences and the AAAS. So for all these reasons, I mean, this is clearly somebody that we want to come listen to. But I think equally important to all of these important positions of power and accolades is the fact that Dr. Carroll is really a storyteller and a conversationalist. And those of you that have had a chance to hang out with him a little bit today, I think I'm seeing a lot of heads nodding anyway. And that's so I think he's a great example to us of the kind of science that we can be doing, the kind of science we should be doing, but also the kind of science and public outreach that we need to be doing. We need to be getting ourselves out into the public, as he said at lunch today, so that people can meet a scientist. And everybody here, whether your mom calls you that or not yet, you're a scientist. And people need to meet you to think about, to understand how you think about the world. And I think Dr. Carroll has a great way of helping to organize those thoughts and can be a great example to us. So I'm really thankful that he could come and talk with us today and share thoughts, both here in this technical seminar and then again tonight at the family-friendly, fun, 7:30 public lecture. With that, we'll turn it over to Dr. Carroll.
Thanks, everybody, and I want to thank Steve, and I want to thank so many people, students, faculty, for the hospitality today. So I'm a native Ohioan, born and raised in Toledo. My first visit, though, to Miami, and this place grows on you really quickly. I truly mean it. What a beautiful place. Of course, I caught it on a gorgeous day, too, but it's a wonderful campus and a wonderful environment. So thank you for trusting an hour of your time to me here today. We'll see. This is maybe my audition for later tonight. I don't know. Okay, so I want to tell you, a science story. Now, I get to talk today about one of my childhood passions, and that alone is a statement of just how lucky I am. And, not everybody's not so lucky. I want to show a clip from a documentary film called It Might Get Loud. It's going to just keep your ear tuned all the way through the clip. I'll have a little more to say at the end. Okay, where are we? This is hopefully it. I'm having a little trouble with the play function on this thing. Come on now. There it is. Now we flunked in rehearsal. There it is.
We were a little local band and we were playing around school dances, and not even the slightest, mildest consideration that would go into something. It wasn't this driving thing that the minute you hit your teens you've got to be famous.
What are your two names? Yours is?
James Page and...
Page House. And you're just learning to play the guitar?
Yes.
From a teacher, do you play anything except skiffle?
Yes, Spanish and dance.
You're doing well. Getting well. What are you going to do when you leave school? Take up skiffle?
No, I want to do biological research.
So, young James Page, unfortunately his dream did not come true. He didn't go into biological research. He had to find an alternate career, one with just limousines and screaming fans and private jets. It's been awful. But we lucky few, we biologists, we're living the dream that Jimmy Page could not live out. So in these trying times, I think it's always important to remember that not all of us, right, are so fortunate. So please keep Jimmy in your thoughts tonight. So my dream, first conceived really when I was a kid catching snakes in the woods and fields around Toledo, was to someday make a living studying these creatures. It took me 40 years to get back to that dream, but I found my way back. So my talk today is in fact going to be mostly about snakes. When you see this creature, and if you're wired like a normal human being, you probably recoil and see something extremely dangerous. But I see an irresistible evolutionary mystery, because just a quick glance at just the external appearance of this snake reveals many morphological features that are novel to this group, from the obvious limbless body to, of course, its famous rattle, or a little hard to see these, the forked tongue, the infrared heat-sensing pits in the front of its face, and well, I guess, sort of much like Donald Trump, when he opens its mouth, it gets even scarier. Fangs and venom. So this creature, it just presents a lot of evolutionary mysteries. And the origin of novelty has been one of the central foci of my lab for more than 20 years. And today I want to talk about how novelty evolves at a couple of different levels. I want to talk about what we know, and I also want to emphasize what we don't know. And the two different levels are really the morphological level, which we've worked on quite a great deal, and we've asked questions about what kinds of genetic changes underlie the origins of new morphologies. But I also want to talk about the origin of new protein functions. When I entered, long before actually I entered the professional biology, there was a dominant idea, and it echoed for decades, about where novelty comes from at the genetic basis. And it came from a short, very influential book by biologist Susumu Ono, who wrote a book called Evolution by Gene Duplication. And right there in the preface of the book, as well as its interior, Ono posited that natural selection merely modified while redundancy created. Essentially, that you need new and spare genetic parts for novel things to arise in biology. And also argued that allelic mutations of already existing gene loci cannot account for major changes in evolution. In subsequent years, a couple other outstanding evolutionary biologists sort of doubled down on this by stating that gene duplication must always precede the emergence of a gene having a new function. And this was the dominant idea. So when I pursued the question, beginning in the 1980s, of the evolution of the animal kingdom, this was certainly the expectation that we all had, that we would see that the sets of genes that existed between different animals had changed and in some way reflected how those animals differed from each other. As it turns out, this was both empirically theoretically and empirically incorrect. And we started to discover that in the 1990s. And I'm going to just give you 3 observations that was really coming a little bit from the early days of Evo-Devo, which is really sort of the comparative developmental genetics of different creatures, but also hints were coming from just developmental genetics of model organisms. And the first hint was that when we studied the body sculpting genes that shape sort of the major features of bilateral organisms, the so-called Hox genes, we were surprised to discover that we knew these Hox genes from the fruit fly. That's where they were discovered. That's where they were really first characterized. And the fruit fly has lots of differentiated body segments in its head and mouth, thorax, a little bit of its abdomen. And not so much in creatures like the centipede or the onychophoran, yet essentially these creatures have the identical set of Hox genes as fruit flies. The explicit, in fact, in print hypothesis was that more recent, advanced, and highly modified creatures would have more of these genes than those with really primitive, repetitive bodies. So we're like, we may need to be thinking a little bit differently that with no new Hox genes came along. It was also discovered, for example, that really the sets of Hox genes don't differ, say, between a coelacanth and humans. Might want to send that postcard down the road to the Creation Museum. Second key observation comes from developmental genetics and beginning to identify the genes that are involved in patterning certain tissues. And I'm showing you just a little glimpse of the functions of a pretty famous gene called sonic hedgehog, first isolated in a trio of labs, most notably Cliff Tabin at Harvard, who gave me this slide. And what you're seeing in these dark areas are regions of the chicken, developing chicken, where the sonic hedgehog gene is active. And at this given moment in time, you see sonic hedgehog active along the neural tube, and it's active here in the posterior of the limb buds, where it's actually going to organize the digit pattern of those limb buds, and later in development in the feather buds. So, question for all you sharp biology students, what do these different body parts have in common besides sonic hedgehog? Why would sonic hedgehog be in all three of these places? And the silence is the correct answer. There's nothing they have in common. There's no prediction you would ever make that the same gene was involved in patterning three such different tissues. And the clue that was coming here, and this is the protein made by this gene is the same in all three of these tissues, that you're patterning entirely different structures in the same organism with the same protein. So what does that mean? And you're using the same protein in really different contexts. So that's that individual genes have many functions was a surprise. We referred to that as pleiotropy of a given gene. And then the third surprise had to do with the logic of the regulation of these body building and body patterning genes. So you're all probably familiar with the fundamental discoveries of bacterial genetics. So the discovery that, for example, the expression of sets of genes, say in a bacterial operon, was coordinated by a single genetic switch, an operator, for example. When the bodybuilding genes were being dissected, particularly by fruit fly biologists in the late 1980s, early 1990s, they discovered that genes that, for example, were expressed in seven very symmetrical looking stripes, that output was the sum of many discrete genetic elements, that individual genes had many enhancers, many cis-acting regulatory elements. Individual elements control individual features of gene expression. That was a really different logic. Well, that logic also said this is how a gene gets used in many different places, that each of those places where a gene is active is generally controlled by a different, discrete cis-regulatory element, or what you might know as an enhancer. So those three observations, when you add them together, that you're not seeing new genes evolving, developmental genes, regulatory genes evolving over vast periods of evolutionary time, that individual genes have lots of functions, and that each of those places where that gene is turned on is controlled by different elements, led to some important evolutionary implications. So that is that individual regulatory genes must have acquired, this is just a simple deduction we'd have to make, they must have acquired many roles and new regulatory elements in the course of evolution. So it wasn't a matter of adding new genes, it was a matter of adding new regulatory elements over the course of evolutionary time. And that means that a process called gene co-option must be widespread. And just so we're all on the same page, let me give you a little formal definition of co-option. That's new uses for existing traits or genes or proteins or genetic elements, whatever that must be. So we can infer that something like sonic hedgehog has been co-opted to be used in the development of, for example, structures that have come along in the course of vertebrate evolution and weren't there in primitive vertebrates. And that's a story, once we started seeing glimpses of this in the early 1990s, that's a story that's just been uncovered again and again and again throughout the animal kingdom. So no, not requiring gene duplication, that in fact much of the physical diversity of the animal kingdom has been built around a common set of regulatory genes that has not expanded greatly in particular lineages. So this then, if co-option's so rampant, how does co-option occur? And I'm now going to save you about a 2-hour seminar and just give you a couple conclusions from a lot of case studies, because I'm mostly going to talk about something else today. And this was a really tough problem to track down. And that's because the origin, finding situations where you can sort of pinpoint, on an evolution, on a phylogeny, and in a particular accessible experimental system, a new gene expression domain, a new morphological feature, that's been a challenge, but it's been done several times. And what we find is that new gene activities, new morphological features, generally evolve by modifications to existing enhancers. So that new features, which may open up new ways of living, which may be related to even new behaviors, that this is evolving by tinkering with these sets of genes and turning them on and off in different ways, and not by really changing the number of genes, the representation of those genes used in development. Okay, so there's lots of examples. Several of those, I think, most of them are coming initially from studies of fruit flies, and for example, how some fruit flies got their spots. But this is I think a widespread occurrence, an explanation that can tell us how can you have basically a very similar set of genes for hundreds of millions of years and yet generate so much physical diversity. That means most of the action is out in the regulatory space of the genome, not in the coding space. This inference kind of echoes some ideas that have been around for a long time. How can we get innovation without duplication? I just want to give you a little schematic that comes from a paper by Bergthorsen and PNAS about 10 years ago. The authors were thinking about this in a slightly different context, but the idea simply is you have a parental gene that has a sort of an original function, maybe a dominant function, but there's some kind of fortuitous minor activities, and we have seen this in empirical studies, and that side activity can become important. And as it turns out, enhancers are really flexible this way. Enhancers are just much more flexible sequences in the ways they can be modified without disrupting their function relative to, say, coding sequences. They don't have a reading frame. I could go on and on about other reasons, but it just turns out regulatory elements can have multiple activities, and those multiple activities may be large or small, and you can tinker with some of these side activities. So this sort of moonlighting model, the idea of how can you add an activity to a regulatory element, that looks to be the sort of action that's going on in the course of evolutionary time to generate new domains of gene expression. This model, though, this side activity model was really being proposed for the evolution of proteins. And what I want to talk about for the rest of my talk today is about the evolution of novelty at the protein level. So if that conclusion sounded kind of odd to you about morphology, that we're really not seeing a lot of action of the addition or subtraction of genes, that may be simply because you're aware that in the course of evolution, all sorts of gene families have expanded dramatically or contracted or whatever you like. And that's absolutely true. And a lot of body physiology, vision, smell, respiration, etc. There's key stories that involve gene duplication and expansion of, for example, of genes and their specialization to different functions. So we know gene duplication is important, but its role and its necessity in the evolution of novelty is not settled. And the reason is that we know from first principles of population genetics is that most probable fate of gene duplicates is to actually degenerate before novel mutations or activity could evolve. And we see this now that we can look at whole genomes. There's a lot of dead genes in our genome that are most likely duplication events where one of the copies has been killed off. Okay, so it was very attractive to think that the simplest way for something novel to happen is genes would duplicate, one gene would retain the old function, the new gene would sort of explore new functional space, and if it found something worthwhile, natural selection would take care of it and you'd get a new function. But that turns out just to be too simplistic, not really reflective of our understanding of principles of population genetics these days, and not well supported by empirical data. So how might we get duplicates? How might we get novel activities? Okay, I'm going to circle back to this later, because what I'm about to tell you is our effort to kind of get into this question of where new novel activities arise. And just to prepare you for what's going to happen is, this is the road we went down but we discovered something entirely in that story, all right? So you're not going to walk out of here with a very satisfying explanation of protein novelty, but that's what happens in research. But hopefully you're going to walk out of here saying, wow, that's pretty weird. I'll remember that. Here we go. And it comes to why we started studying snakes, which goes beyond childhood passions, but not much farther than that. It's a midlife crisis, folks. That's what I'm Steve has arranged this intervention, and I really appreciate you coming back to my homestead. Okay, why snake venoms? You know, if I had to put this in a grant, well, it's a novel, obviously, key trait, really important to the lifestyle of these snakes. Think about it, these venoms are generally mixtures of proteins that are working in some sort of combinations to do the job of, for example, subduing prey, even starting sort of the digestion of prey. Venoms are biochemically very diverse among families of snakes and between species, and I'll show you even at the intraspecific level. And I also knew, and this is just the lore of a curious kid reading whatever was published out, well, not published, this was generally popular books about snakes, that there's a lot of natural history out there that, you know, because these are not common, you know, subject matter for sort of, you know, genetic research or things like that, that there are just mysteries out there that people haven't poked into hard enough. And I'll talk about a couple of those today. So that's one of the reasons to get into these snake venoms. And we know that these snake venoms, like rattlesnake venoms in particular, they're an arsenal of proteins. Serine proteases, metalloproteinases, things like bradykinins, lectins, and phospholipases that work as both muscle toxins, myotoxins, as well as neurotoxins. So you have this whole concoction. You're sort of thinking, you know, how are snake genes somehow weaponized to create venom? You know, are these just normal physiological genes that were somehow by, you know, being turned on in a venom gland, they become, you know, toxins, or are they modified in some way that they're a novel protein activity that didn't exist before in snake history? So I'm going to focus in particular on one type of toxin known in rattlesnakes because it looked to us to be the most promising inroads into this question of novelty, where do things come from? And it has to do with a neurotoxin. It's a heterodimeric neurotoxin, meaning it's a neurotoxin made up of two protein chains. Each of those chains is is based on a phospholipase A2 enzyme type structure. But here shown in the protein model, these two chains, one is an acidic chain, one's a basic chain, they come together to form this heterodimer. And this is a potent toxin. This will, in humans, for example, it will halt your respiration. This is not a toxin you want to encounter in some of the snakes in the United States, for example, that possess it. This makes the snakes that have this in their venom are, to humans, a little more dangerous than just your average rattlesnake. And the way I put that to you, what we knew was that only a subset of, for example, North American rattlesnakes, or North American and Central South American rattlesnakes, have this neurotoxin. In fact, it looks so restricted in its distribution, looked like a promising case of novelty. Sort of the more famous snakes, if you've heard of things like Eastern diamondback rattlesnakes and Western diamondback rattlesnakes, they don't have this neurotoxin. They are largely hemorrhagic venoms, and that hemorrhagic activity is especially promoted by metalloproteinases in the venom that break down tissue, break down blood vessels, cause a lot of hemorrhage, things like that. So this neurotoxin was something different. So we thought, okay, where did these rattlesnakes get this neurotoxin? And so to start that, we asked, all right, what's the genomic origin of these neurotoxins? How are they encoded in the genome? And there hadn't been any work done like this on rattlesnake genomes, so we did it. And you're going to see a lot of these maps, so I want to make sure I kind of keep everybody on the train here and give you some orientation. So there's a nomenclature here, but the ones in color are sort of snake-specific phospholipase A2 genes. The genes, well, I'm kind of lying, they're not just, the ones in the bright colors. The ones out here in the flank, you'll find in every vertebrate, okay, including us. But these guys are expanded in snakes. In other words, these are not genes that you'll find in outside of snakes, and you won't even find them mostly out of the group that includes rattlesnakes. And what I've circled here are just a shorthand for the two subunits, the basic subunit encoding gene, the acidic subunit encoding gene. And this is what the gene complex looks like at the genomic level of where these toxins are encoded. So they're nested among a bunch of other phospholipase A2 related genes in a part of the genome that has even other members of the phospholipase A2 family. There's lots of these genes in our genomes. But in snakes, these guys are the ones of interest. So we asked a really simple question. There are the genes for those subunits. What does this complex look like in snakes that don't make neurotoxins? Okay, so I'll show you the comparison here. So the neurotoxic guys in the center. This is a rattlesnake called the Mojave rattlesnake or Cordylus scutellatus. And there are the neurotoxin subunits. And if you look at, this is the western diamondback rattler. This is the eastern diamondback rattler. Those genes are just not there. So this looks to us like a nice case of novelty, right? Present in this guy, this potent neurotoxic producing snake, absent in those two guys. But to make that inference, you have to know something about the evolutionary relationships among rattlesnakes and of the neurotoxic or non-neurotoxic status of snakes. And when we map that onto a phylogeny of rattlesnakes, we got a little bit of a surprise. And the surprise is that the neurotoxic guys, there's a clade of neurotoxic guys shown here in blue, and sprinkled among them are some snakes that are not neurotoxic, such as the eastern diamondback that I just showed you, the western diamondback here. Okay? So this is a clade, there's a much bigger group, well, a somewhat bigger group that includes all rattlesnakes. So these neurotoxic rattlesnakes, they cluster in a clade, and again, that looks to us like, okay, well maybe something went on here in terms of the origin of these neurotoxin genes. But really, when we map this neurotoxicity and think about how many times neurotoxicity must have been gained to explain that pattern, it's not a very parsimonious interpretation. The most parsimonious interpretation is that neurotoxicity, rather than being gained among individual species, that it was present in the last common ancestor of these rattlesnakes and lost several times. So with respect to this group of snakes we're studying, it's not that neurotoxicity is a novelty, it's just that the loss has occurred in particular lineages. So we sort of end up turning for 40 or 50 years of herpetological thinking about these rattlesnakes upside down, but it's inescapable from mapping the presence of the neurotoxins or the presence of the neurotoxin genes onto a rattlesnake phylogeny. Okay, that might make you wonder from an ecological point of view. I just told you what a great little weapon this neurotoxin is, but yet some snakes have lost it. Maybe we'll take that question, maybe we'll hold that in your head for a minute. Well, maybe we'll talk about that just a little bit later, because if you think this is weird, the story's going to get weirder. And I might as well tell you the weird part before we deal with why would snakes lose something that was a potent toxin. Okay, so the most recent common ancestor is neurotoxic. The genes were lost. And they're not, when I say lost, if you have any genomic experience or you know about sort of human genomes, lost means lost. They are gone. There is no trace of these genes. That's an unusual pattern because a lot of genes, when gene activity is lost, there's a pseudogene in there. There's some mutations that have crippled the coding information in the gene, but the gene is still represented somewhere in the genome. Not the case here. These genes are wholesale gone. And I'll get to the how, I'll get to the why maybe later in the talk, but how these genes are lost, we saw that this gene complex in particular was just riddled with repetitive sequences. And we suspect that what those repetitive sequences do is they facilitate a non-homologous recombination, non-allelic homologous recombination among the non-allelic homologous recombination, which means if repetitive elements from different parts of the chromosome cross over among them, you're going to get deletions and also expansions of the complex. So this is just a picture that we could find. There's lots of smaller repetitive elements, but there's big stretches of repetitive elements in the genome, and this is just to give you a sense of where those things are. And we know if you recombine between, for example, this one and that one, it's pretty easy to explain how you could lose all three genes. Because while I didn't quite point that out to you about the differences between these species, not only are the neurotoxin genes absent from these other two species, the ones that are present up here, but there's even other genes that are present up here and absent down here. So we're seeing other things that have disappeared, adjoining genes that have disappeared. And I think the other kind of satisfying hint that this is a pretty dynamic situation in these snakes is we just happen to look at 4 individual western diamondback rattlesnakes to try to make sure that the structure of the complex was as we thought it was. And in one of those snakes, we found a copy number variant. That there was an addition of 2 genes and some other flanking sequence that had been added out here to this part of the complex. So this is, we'll call it the wild type that we saw in 3 individuals, but in the 4th individual, we see an expanded complex. So if you have this region, this sets of genes, and I can tell you, for example, in the western diamondback, this gene here highlighted in red, this is a potent little myotoxin. It makes the bite of the western diamondback quite painful. So that's an important gene to the way the western diamondback lives, and that gene is not present, for example, in the neurotoxic guy I just showed you. So these other genes are important, but what's happened in this individual is you get added additional phospholipase A2 genes. So the picture I want to give you is when riddled with these repetitive elements, it essentially facilitates the expansion and contraction of these gene complexes. You have a molecular template for the expansion and contraction of these gene complexes. So we think there's rampant, well, from an evolutionary point of view, a lot of gene addition and loss going on in this complex over a relatively modest evolutionary period of time. In the case of these rattlesnakes that I just showed you, their divergence probably spans 5 to 7 million years, which is sort of a similar distance as humans, chimps, and gorillas. For a lab that had basically studied gene complexes that hadn't changed at all in 500 million years of arthropod evolution, this kind of shook us up, okay? So it's in a good way, all right? So really dynamic complex. It also is kind of fascinating to us that there might be snakes out there with, you know, novel genes, et cetera. But this pattern of loss is certainly not what anybody expected. And what we understood as we compared the structure of these complexes with other snakes is that this group of genes I'm showing you here, sort of in the center of the complex, these expanded in the lineage that gave rise to rattlesnakes, a group of snakes called pit vipers. And if you look at other snakes like cobras or pythons, they generally have only 1 copy of this gene family. And so what's happened is in a rattlesnake ancestor, this thing expanded to about 7 genes. And in individual lineages like that led to this Mojave rattlesnake, or the eastern diamondback, or the western diamondback, is you've had sets of adjacent genes deleted. All right, so very dynamic in terms of the sets of genes. Okay, so this is the part where, you know, it took us a while to get used to that. So you have a very deep origin of these genes, and then what's been going on recently is loss. And we were pondering things about that loss, but we wanted to turn to a mystery in the natural history of these snakes, as is promised you. This is at least to us where it got a little weirder. And that has to do with the observation in herpetological lore that there are within certain species individuals that are neurotoxic and individuals that are not. And they're essentially hemorrhagic. Same species, in fact, even in the same county. Okay? So there are 3 occurrences. It's funny, yellow decided not to show up on this, but we'll show them. Okay, so we'll start. Different projectors, who knows? Is it on my screen? No, that's still there. It's there on my screen. I was worried that Bill Gates deleted yellow from PowerPoint. But that's an upgrade you have to pay for. Okay, so out here, it's where this blue dot is, but basically what's outlined here is the range of a snake called the Southern Pacific rattlesnake. And where the blue dot is a population that's known that's neurotoxic. So the snake is largely not neurotoxic, but there's a neurotoxic population very near Riverside, California, in case you go on vacation. In this blue swath here is this Mojave rattlesnake. It is largely neurotoxic, but there are non-neurotoxic populations and individuals in these Arizona counties marked with these red dots. Okay, so inverse situations, predominantly neurotoxic with subpopulations that are not, predominantly not neurotoxic with subpopulation that is, and then out here the timber rattler. I'm looking at Ohio. I don't know, guys. Any timber rattlers around here? I don't think so. That's said with some confidence too. There's always people who know their snakes in every biology audience. So maybe we get to talk later. Anyway, but there are timber rattlers in lots of the eastern half of the United States. And here's the weird thing: these blue dots mark occurrences. So timber rattlers again are predominantly hemorrhagic, but these blue dots mark various counties. South Carolina, Arkansas, Louisiana, you can see various places where neurotoxic individuals have been identified. So we asked, what's up with this? And, that sounds like an easy question to ask, but I gotta give some shout out. I'll talk a little bit more about the team that was involved in this at the end, but this level of comparative genomics where you actually must, because so much is going on in these gene complexes, we must have really extremely accurate sequencing for these snakes. So this took a while to sort out, to get individuals representative of different types, to look at their phospholipase A2 complexes, and to figure out the story I'm about to tell you. So I'll start with the Mojave rattlesnake. This is the sketch of what I showed you before, the cartoon of the gene complex. The B2 and the A2 genes are the neurotoxin subunits. So what does the non-neurotoxic individual look like? It looks like that. Okay, so just stare at it for a second. This is a what the hell thing in the lab. This is where you don't believe your data when it's coming in. What did I do wrong? Did I screw up the samples, etc.? Remember, individuals are members of the same species, and what we're seeing here is this guy up here has 3 genes that this guy doesn't, and this guy down here has 2 genes that this guy doesn't. So at this gene complex alone, there's the difference in presence-absence of 5 genes. Same species. Don't worry about the taxonomy, etc. This is solid, okay? 5 genes different between these 2. Okay. So we said, what's going on with the Southern Pacific rattlesnake? Similar, different in detail, but again, a situation where the neurotoxin genes right there, the B2 and the A2 are there. They're not there in the non-neurotoxic guy, so the in-between, the gene in between is also missing. So you see a similar pattern of fewer genes. They differ here by 3 genes between the neurotoxic form and the non-neurotoxic form. And what's going on with the timber rattler? I won't make it too complicated for you here. Just pay attention to the upper lines. It turns out this little guy was heterozygous in a funky way, which is a fun thing to find out after $30,000 worth of sequencing. Anyway, its complex is different, and it has, not only does it have the neurotoxic subunit, it actually has an extra copy of the basic subunit. So these other guys come down to the non-neurotoxic individual, it's missing a whole bucket of genes. So again, we're seeing in non-neurotoxic individuals of the same species, fewer genes. Back to the Mojave rattler, it's a weirder situation because there's actually genes in the non-neurotoxic guy that aren't in the neurotoxic guy. All right, so how could we explain this? What are the evolutionary mechanisms that could give rise to this? Well, there are 3 we could come up with, and if you could come up with a 4th, I'd appreciate it because this hasn't gone through review yet. We first want to know, could there be, now remember these snakes are, the geographic area of 2 are close, but 1 certainly not overlapping. So we want to know, has this happened essentially independently at least 3 times in these different species? And are you evolving the non-neurohaplotype within the species? So for 2 of them, we see that the non-neurohaplotype, haplotype meaning the set of genes on a given chromosome, is reduced relative to the neurotoxic form. So you could imagine that you've just shrinkage, right? And I'll show you there's a little problem with that. Another possibility a little more complicated, and you may not be too familiar with these cases, but if you're polymorphic for a given gene locus, and think what will help you because this is a true story, in humans, for example, for ABO blood type, and the species splits to give rise to 2 descendant species, well, what's going on in those descendant species? They may in turn also be polymorphic, right? And so the polymorphism may be something that's been inherited through time, through the process of species formation, through lineage splitting. And so we envision the possibility that maybe some ancestral rattlesnake that gave rise to 1 or more of these species was polymorphic. And what we're looking at is the doling out of multiple haplotypes, ancestral haplotypes, through speciation events. So that would be different than these independently evolving. What you're basically doing is you have multiple haplotypes in an ancestor, and you're sort of parsing them out in populations differently. And then the non-neurotoxic things like eastern and western diamondback, well, they've got only the non-neurotoxic type haplotypes, right? And they're homozygous for it, and that's that. So if you have snakes that are polymorphic, this might explain the polymorphism. Some snakes are monomorphic because basically they've coalesced to just have one kind of set of genes. And the last possibility, which you probably already thought of, is perhaps hybridization. maybe you're getting some swapping of genes between rattlesnakes, for example. And it could go in either direction. You can look at whichever direction you want. If a neurotoxic guy and a non-neurotoxic gal get together, okay, genes are going to get exchanged from different species. So what do we know about this? Well, just think about the independent evolution. We don't think that could possibly explain this situation of the Mojave rattler, because this sort of set of neurotoxic genes, you can't derive this bottom haplotype from this, right? Because there's genes here that are up there. So that's got to have it's got to have a different explanation anyway, not that simple independent evolution explanation. And also, these haplotypes are different than the haplotypes that exist in the other species I showed you that are polymorphic. So, a lot of genetic variation going on here. And what's weird about that variation, most variation biologists study is variation at individual genes, sort of simple nucleotide substitutions, right, at individual genes. This is variation at the level of presence and absence of entire genes, and in this case, multiple genes. All right. Anybody having a good time? Whatever. I am, so that's what counts. Because to us, it kind of got better. So you might say, if you're running around in your head, you might say, okay, these snakes, this is weird. The neurotoxin is powerful. Why would individuals not have it? Why would some species not have it? If their ancestors had it, etc. Well, be thinking about it. These snakes are perfectly capable of killing their prey, or they wouldn't exist as anymore as species. So let's talk about the other genes that might be important to the lifestyle of a snake. And so we invested a really big effort to chase down the metalloproteinase genes of these rattlesnakes. And the reason why it was a big effort was because these complexes were hard enough. These complexes are just evil, okay? Big set of genes. Whole suite of tandem arrangement of metalloproteinase genes that encode, and if you look at the legend down here at the bottom, a lot of these genes aren't expressed in the venom gland. Okay, so that's also true of those phospholipase A2. Some are expressed in venom, some are not. But we have to understand what's going on with all these genes to understand the evolutionary history of the genes used in venom. So you can see some of these genes, for example, are highly expressed in venom. So in the Mojave rattler that's not neurotoxic, it has metalloproteinases, 2 of which, for example, are highly abundant in the venom. I think they're doing most of the work in the venom. So we said, okay, what's going on in this gene complex in the non-neurotoxic individuals? What the heck? It's an 11 gene deletion. It takes out the predominantly expressed genes. Okay? 11 genes. So really, at these 2 gene complexes, they differ by 5 genes, presence absence at the PLA2 complex. They differ by 11 genes at this complex. And we looked at multiple individuals just to make sure we weren't being led astray. All right. It's a strange way for genetic variation to be carried, or for sort of biological variation to be. So what's going on? So we looked in the same way at this Southern Pacific rattlesnake and its metalloproteinases, and here was a reward. Again, this is a lot of sequencing. Start with the title, don't look at the bottom detail for a second. And the title is this: the metalloproteinase complex with that 11 gene deletion, the exact same haplotype is in the Southern Pacific rattlesnake. The exact same haplotype that has the exact same sequences at the border of the deletion, and it shares single nucleotide polymorphisms between the 2 species. This is just a lot of evidence because people ask for it sometimes when I talk about it. But the point is, that's telling you that the same metalloproteinase gene complex haplotype is in 2 species: the Southern Pacific and the Mojave rattler. That is a red flag of either 2 possibilities, though. Can't sort between them yet. One would be that their ancestor had this haplotype and they both inherited it, or they've swapped haplotypes. Haplotypes have been exchanged, hybridization between the species. It turns out we just need other data to sort between those possibilities. But whichever the case is, you either have an, I'll call, an unusual pattern of inheritance of these gene complexes from ancestors, or you've got hybridization going on, creating novel genetic combinations. So what I told you about. So that we at least would say, we think this independent evolution of alternative haplotypes is not going to be the fertile ground. That either we're dealing with a trans-specific polymorphisms and/or, because both could be going on, hybridization between species of different phenotypes. And that's generating diversity out there in the world. What's extremely weird is that this diversity is in the form of presence of absence of entire sets of genes in 2 gene complexes. And we were really puzzled, has this ever come up before? So the reward to me, when I said I was going to tell you all about novelty, which I'm not, is that we just, as a geneticist now, I'm really puzzled. I started asking all sorts of colleagues who worked on all kinds of organisms, have you ever seen anything like this? And maybe there was some stuff in microbes, but for vertebrates, this is weird. But one case came up from the literature in terms of precedent. And that has to do with these genes, these killer cell receptor genes that work with histocompatibility proteins on the surfaces of adjoining cells and are involved in both immunity and, interestingly, in reproduction. For example, things that go on in the placenta. And there's really striking genetic variation among primates, including humans, in the sets of genes here. So I don't want to, this is the nomenclature of those genes. I've already killed you with all this stuff. But the point is that individuals, for example, individual humans across the globe, differ in the sets of these killer cell genes in terms of the entire, the haplotype sets that different humans have inherited. And, that is interesting to us that we can find this kind of genetic variation anywhere in another vertebrate. But a lot of work's been done on these genes by biologists of various stripes. And their sense is that the reason for this genetic variation has to do with host pathogen evolutionary arms races. That there's selection for these different haplotypes, maybe even some selection against certain haplotypes, depending upon how these things are interacting. But that's been a driver of this genetic variation, such that you have these presence-absence variants. And because this is involved in immunity. So we thought, well, that might be informative, because now let's talk about why this variation might exist. So what we've got here is we've got extremely divergent haplotypes in 2 toxic gene complexes that encode alternative venom types in rattlesnake species. So the haplotypes, just in case you're wondering, they appear to segregate independently. They're not linked. Okay, so what's going on at the PLA2s is being handled independently of what's going on at the metalloproteinases. And we wonder, just as the inference is, a reasoned inference is being made about the killer cell genes, is that this could have to do with sort of an arms race with prey. That across the geographic ranges of these snakes are eating, depending on their body size and prey availability, they might be eating, mammals, birds, reptiles, so, mice. I hate to say voles after my lovely vole talk today. Lizards, this sort of thing. And there may be both different prey availability, but there also may be different prey susceptibility to these venoms. We know in certain parts of North America that some prey, like California ground squirrels, Mexican ground squirrels, have evolved resistance to rattlesnake venoms. We know that some predators that like to eat snakes, like opossums, have also developed resistance. So we could imagine that it could be that either susceptibility or availability is selecting for different haplotypes across the geographic range of these snakes. And that this genetic variation is then being maintained by balancing selection. So more work to do to understand the mechanism, whether this is ancestral variation that's been around for a long time in snakes, is being sorted in different snake lineages. More work to do to see to what degree these snakes are hybridizing or have hybridized with one another and created more genetic diversity. But I guess the last take home is you never know what you're going to find when you start a research project. As a geneticist, this is really, to me, really satisfying. I hope it is to you, because I just ate 50 minutes of your life. But I think in a larger context, when I first started telling people that the fruit flies are gone from my lab and we're going to work on snakes, I got that look of, okay, he's done, he's lost it. And that may be true, but nonetheless, I did not expect to uncover a phenomenon like this so early in our research that is so rare as far as we know, in terms of the behavior of vertebrate genes. So we found that rewarding. But we wouldn't have found it without two key ingredients. And one key ingredient are people on my team who took the leap of faith to do work unlike anything they had done before or had been done at all, including climbing the learning curve of vertebrate genomics and vertebrate comparative genomics. In fact, had their fellowship rejected from the NIH, said don't do this as a postdoc. Genome sequencing is too complicated. Not kidding. Shit, NIH. Not the first time. Not the first time, folks. I'm sorry, that just came out there, Steve. It's after 5 o'clock. It's after hours. Okay, so the brave people in my group are Noah Dowell and Matt Giorgiani. There's Noah. There's Matt, with tremendous help from Vicki Kastner and Sam Griffin and Jane Seelig in my group. But there's no way we could do this work because, no, I do not have these rattlesnakes in Wisconsin. I don't even know if there's enough biosafety forms to get them, okay? But what's great is the University of Texas, or Texas A&M, Kingsville, has the National Natural Toxins Research Center run by Elda Sanchez. And Elda Sanchez and Mark Hockmuller, that are collaborators on this, hosted Noah and Matt, and just been great resources as all the twists and turns have happened in the course of this project. So I want to thank them and thank you for listening.
A few questions?
Yeah. So, this is normal. He actually wrote something about snake in his the essay written in Japanese, and he said the snake with strong venom doesn't make sense because those snake has really strong venom to kill the prey, but at this same time they have behavior to tell that we are scary, we are venomous.
Yeah.
But if that venom is so strong and no one gonna survive, no one to survive to tell that snake is venomous and dangerous.
Yes.
I wonder if these allele, like less venomous allele, or like the individual, have something to do with teaching the
Teaching. Okay.
or prey or.
All right. So I had not having read this essay in Japanese. Slight disadvantage.
I was thinking.
So let's think about these things. Thank you. That will really help a lot. I'm sure it'll be a beautiful decoration. We know about the LD50s of snake and their prey, all right? And so generally, the amount of venom delivered is overkill to killing, but there's things in these venom that also, for example, they tend to be like paralytic to the back limbs, and so the prey can't get away, because generally these snakes are ambush predators, right? The prey goes by, snake goes bam, it's got to use its heat sensing to track sort of the dying animal down and then eats it. So, that sort of behavior and sort of the prey subduing role of venom, I think, is well understood. Now, the warning: these snakes evolved, in probably at least the last 12 or 15 million years in North America, out on a landscape that had a lot of big mammals. I think that rattle says, don't tread on me, right? Because they're not going to take, they're not going to eat a bison or anything like that. But when stepped on by a bison, it's a bad day for a rattlesnake, right? So I think that rattle is, evolved a long time ago with a fauna that would have a lot of large mammals. So I think that's simply self-defense. Now, from a human point of view, obviously they haven't been around humans long enough to evolve, much. If they had, they would slither a lot faster across roads, because that's where they're really losing the game, and at a great disadvantage if you've ever been out on the roads in Arizona or someplace like that. So so that I think is the simple logic, is that the rattle is, you're big and bad for me, I can make you sick, maybe even worse, so please, stay away. Now, there are some standoffs. I know there's some behavioral work done on like ground squirrels, versus rattlesnakes, and ways they sort of interact with each other. I don't have a deep enough knowledge to sort of say much more about that. So when you've got a situation where the prey is potentially subduable, but maybe it's more of a borderline case for the snake. These two warning, I don't know, between the warning system of get away or hey, maybe I'll eat you. I don't know how that interplay is, but I think for large mammals, it's a pretty clear case. They're not subduing large mammals, and they don't want to be stepped on. Does that make sense?
Yes.
Do you want to write that in Japanese?
Yeah.
I can publish it.
No, the part I wonder is the evolution of these less venomous animals. So I thought maybe these.
Less venomous? You mean?
Yeah.
Oh, you mean these guys that are less the neurotoxic being less venomous?
Yeah.
Well, I mean, there's different games to play. So, if we just measure as biochemists and we say, what's the difference? Maybe the LD50 is about 1/3 that of the hemorrhagic venom, but you're overwhelming the prey anyway. So, overkill is overkill, right? So from our point of view, it matters because for a human patient, we're often kind of right on the borderline of is this a potentially fatal, or how sick can we get from a particular snake bite? We're pretty big, we're 70 kilograms or more. And so you would really like to know, did you get bitten by a neurotoxic or a non-neurotoxic? Because the symptoms are going to be really different, right? You're going to worry about respiratory arrest if it's a neurotoxic snake. So a clinician wants to know that. But I would not consider these more or less venomous. From the viewpoint of a small rodent, they're venomous. Yeah.
For the two species that lost the genes, what are the intergenic regions? Like, I'm assuming the coding regions are very highly conserved. What are the intergenic regions like? Are they significantly different, or are they?
We see shrapnel in there. Like, we see exon fragments and pieces of repetitive DNA in there, such that we can sort of find anchor points that align these different complexes. So that would tell.
I'm thinking more along lines of the 5' and 3' UTRs and these things. Are they?
They're gone.
Okay. Oh, really?
They're gone. Yeah.
Wow.
Yeah. Yeah. So, I'm saying that it's in general true. It could be possible there's a case where some 3' or 5' exon is still remaining, but generally they're gone. Yeah, nothing there. So these are pretty big deletions. We of course don't know how many steps the deletions are taking, but we can picture these deletions taking place, taking a couple genes out at a time. Yeah.
So when would you consider this the venom being novelty? Like the common ancestor, how?
Yeah, so you have to go back in snake history to say the novelty. So what we do know is the sets of genes I was showing you about, the PLA2s for sure. That set is expanded in the group known as the crotalids, the pit vipers, and relative to outgroups like boids and elapids, which include things like the cobras. So it's in deeper time that this complex was expanding. And that expansion, the pit vipers are an old world group. So this happened, first evolved in most likely Asia, at least somewhere between 20 to 30 million years ago. And then the ancestor of rattlesnakes came to North America maybe 15 million years ago or so, and had these genes. So it's a deeper time issue. If we did more sampling outside of the rattlesnakes through the crotalids, I think we could reconstruct a picture a little bit more of how this complex came together. What's interesting to us is that these phospholipase A2s, for example, they're used in digestion, right? They're a common pancreatic enzyme, things like this. So it's interesting to us, what was the forerunner of these toxins being used in a non-poisonous snake? And we suspect maybe something in saliva, maybe an oral gland, maybe pancreas, something like that. And somehow, maybe the first step is the expression of that enzyme at high levels in some structure that can be, that can inject something into prey. Then we think, to get the heterodimer, we think is good bona fide protein evolution must have happened because these other members of the gene family don't heterodimerize. So that we think that may be something that was, toxic on its own as a monomer or as a homodimer. That something happened that brought the acidic and basic chains together. So we think we could do some reconstruction of that history with deeper sampling and get at, for example, where did the heterodimer come from? I think we could get there. But it just turns out where we started was really all these loss events, and then discovered all this weird variation. So I think it's traceable, but we haven't, and nobody else has sampled the taxa you'd want to sample to start to reconstruct the earlier history of these genes. There's sort of, we know the outgroups don't have them, and we know some more contemporary species that do have them, and we need to get sort of into Asian crotalids and reconstruct some of that. But it's a time and resource consuming process to do the full-scale sequencing of this. Yeah, go there first, yeah.
So based on the pattern you showed us, with the geography and everything, like to me the hybridization seems very plausible.
Yeah.
I would also argue for drift over selection, but my question is, you could probably easily test it by predicting who would be the hybridizing species. So with your Helleri scutulatus, that's very logical combination.
That's right.
With the Horridus, it's probably a diamondback, right? So can you do a similar comparison between the non-neurotoxin Helleri and the nearby
Yes.
Adamanteus? And if you get a match, then that would be a pretty convincing case of.
Yeah, I think you're on the right. Track right there. The scutulatus and the Helleri are very close to each other today, and who knows where the ranges were 10 or 12,000 years ago, right? So it's suspicious that they were sympatric, and this could have happened. So we are looking for the genomic signature of process hybridization between those two. It's not, just to follow up to your point about the timber rattler in the eastern United States, it's, that situation's not so clear. That we don't really have a candidate for how that came about. And so while the hybridization I think might be the most immediate thing for us to search for the signal in, and we kind of should know how to do that. If trans-specific polymorphism has also gone on, which I think is very likely, because these snakes are species, it's been a radiation. Rattlesnakes, depending on how you want to count them, let's say a few dozen rattlesnakes in a relatively short period of time. Pretty obvious that if you have imagine what happens if the timber rattler splits into species today. Well, you've got a polymorphic snake giving rise to polymorphic descendants, right? So I think trans-specific polymorphism is a really likely phenomenon going on as well. So we got to keep our eyes open to that. I don't think we shouldn't get. I think we smell hybridization as you do. I think between Helleri and scutulatus, but we don't think we shouldn't get too narrowly focused on that because I think it's very likely, given the amount of variation in these snakes, that variant species are giving rise to yet more variant descendant species, right? And there's one more here, and before. Last one?
There are some poisonous or venomous lizards too, aren't there?
Yep.
And are the venom genes in the lizards related to these venom genes in the snakes?
Nope. Great things snakes have done. In fact, phospholipase A2s are also involved in toxins in the old world and things like that. It's different. These are different genetic events. So snakes have come upon using these sort of ancestral genes for toxins more than once. Isn't that fun? So there's a lot to study. It just turns out not a whole lot of folks are in the business of this yet. Certainly at this sort of genomic origin level. There's been decades and decades of work on the venoms themselves. Some work on individual genes, but I don't think until we looked into the genome and realized how big these families were and how evolutionarily dynamic they were, that we then knew what kind of research methodology we have to use to do those comparisons. So, yeah, lots of ways to become venomous. Isn't that grand? All right, thanks very much.
Regarding this talk, we have a reception upstairs, so you're welcome to join us for food and further discussion. And then, of course,